Hopefully you had the chance to go over the videos for membranes. All right? Those 4 videos, again, cover the entire chapter, and we're going to cover the chapter today. Okay? Point of the chapter is to go through the structure, the current model structure of cellular membranes, eukaryotic cell membranes. All right? And then also go through their basic function in terms of transport, moving things in and out, and the different processes involved in that. Diffusion, facilitated diffusion, osmosis, and active transport and bulk transport in terms of endo-exocytosis. Okay? We will go through all of those today. All right? All of that today. First off, membranes. What are they composed of? Danielle. Phospholipids. So let's talk about the structure of a phospholipid. Right? We know that phospholipids are composed of what? Hydrophilic heads, hydrophobic tails. What makes up the head? A phosphate. All right, so we have a phosphate, PO4, that's attached to what? So here's your fatty acid chains. What do those fatty acids link to? There's a molecule in between there. Glycerol. All right, so we have glycerol molecule, which is a 3-carbon sugar. All right, to one of those carbons, a phosphate is attached. To those other 2 carbons, 2 fatty acid side chains. All right. Are phospholipids saturated or unsaturated fatty acid chains? And what's the difference between those 2? Saturated is all hydrogens. Unsaturated has the presence of what? It's unsaturated, has double bond. Okay. What are typically, phospholipids are typically what? Are they saturated or unsaturated? They're typically unsaturated. Why are they unsaturated? Amy, Joe. Sorry. Because they're liquid at room temperature. All right. But why the fatty acid chains in phospholipids, why do they need to be unsaturated versus saturated? Mike.
They need to be straight. They can't have the bends in them.
So they need to have the bends. You were right. It has to be the opposite, right? So let's erase this and let's just do it like this. So this being a fatty acid chain that has all a saturated fatty acid chain, okay? And then in blue, we're going to have an unsaturated fatty acid side chain. Because the double bond, which is right here, right? The double bond, that's a bond, sorry. All right, it causes a kink in the tail. All right? When there's a kink in the tail, that now prevents other phospholipids from packing in too tightly. All right? So that's the structure of a phospholipid. How are they arranged in terms of a membrane? In a bilayer. All right, so here's our phospholipid head and our hydrophobic tails. And the bilayer is arranged so that the hydrophobic tails are pointed towards one another. Okay? That's the phospholipid bilayer structure of the membrane. What else is present in the membrane? Proteins. All right, proteins. Okay? We call this the fluid mosaic model. And we had talked about that before when we talked about the structure of phospholipids. Fluid mosaic model includes phospholipids and then proteins that are somewhat studded or found attached to these or embedded within the phospholipid bilayer. Correct? Yes? No? Maybe. All right? Give me an example of a protein that you know is an integral membrane protein, meaning it's integrated into the membrane that we talked about last week. What? There was a particular name of a protein that I'm looking for that it's that's a type of protein. Yes, it is transmembrane. It spans both sides of the membrane. But there's a type of proteins that were membrane proteins that have functions both on the extracellular side and on the intracellular side. Integrins, right? And I said they came with. So you are familiar with at least one type of specialized population of proteins that are integrated into the membrane. Okay? And then also on that you have proteins that are on the periphery. Right? These are peripheral proteins. They could be both cytoplasmic or on the cytoplasmic side or on the extracellular side, face of the membrane. All right? Questions on the fluid mosaic model by itself. So the membrane is a mosaic. It's a collection of different parts. Phospholipids, proteins, there's even some carbohydrates that you can include in here. You know, that carbohydrate chains that come off proteins, right, what we call glycoproteins. All right? So it is a model, it's a collection, it's a mosaic of different parts that come together to function as a unit. All right? The fluid part of it says that this is not stagnant. It's not set in stone. These proteins do not stay in one area. The phospholipids do not stay in one area. There is movement about them. Okay? So a protein gets embedded into the membrane and it can move across the membrane. There can be collections of it. Right? They can come collected into one area, we call recruitment, or they could be dispersed, right, and move throughout. Questions on that? So the fluid mosaic model. Here's a picture of your book showing your structure of phospholipids. Right? You'll see here this double membrane in here, very tightly packed. Okay? The closer the phospholipids come together, the more tightly packed they come, the more rigid the membrane becomes. Okay? So, to prevent that, another aspect, another constituent or component of the membrane system is cholesterol. Okay? Cholesterol and its structure right, little lipid structure that you have there. Cholesterol, okay, actually embeds itself. Oh, I can't do it now. Anyways, will embed itself into the membrane. Okay, it will embed itself and stick in the hydrophobic region of the membrane. Okay, by sticking in there, it prevents the membrane from becoming too tightly packed and helps maintain the strength of the membrane and its fluidity. Right? Because as the membrane becomes too packed together, it becomes rigid and can be broken. I remember. Okay? Here's what you look at in terms of your unsaturated tails prevent your packing. Right? They leave open space. Saturated tails pack tightly together. Right? And then cholesterol embeds itself into the membrane, right, to prevent that from occurring. This helps to maintain fluidity. Okay? Temperature has a lot to do with this. All right? So as cells freeze, why do cells break when they freeze? What happens? Why do cells break when they freeze? What happens to water when it freezes? It expands. So why do cells break when they freeze? So what happens is temperature goes down and you move towards this configuration more than this configuration. All right? You get more tightly packed because of the temperature. The interior of the cell is made up of mostly water. It expands because there's no more stretching of the membrane breaks, cells freeze, right? And membrane breaks, cells die. But in research, in biomedical research, we freeze cells all the time. And we store them for years and years and years in liquid nitrogen, right? And then we bring them up and we can make them grow again. How does that work? How do we freeze cells that are 98% water and keep them going? Do we keep them in water? No. Would researchers have to add something to the media or to the freezing medium to prevent membranes from breaking? Yes. We added a lipid called dimethyl sulfoxide, DMSO, which is a cryoprotectant. Prevents this from happening. It prevents the membranes from breaking. So even though the cells freeze and you get an expansion of the cytoplasm, it doesn't cause a break in the membrane, and it helps keep cells alive. All right? Here's a picture of a protein. All right, that's embedded in the membrane. This is a transmembrane protein. It spans both sides. Note that you see parts of this. All right? What you see here is a globular structure of the protein, right? Which means it's which level of protein structure? Tertiary. All right? What do you see in terms of those ribbon structures up here? What are these things? The helixes, which are parts of what type of protein structure? The secondary structure. Well, it's a globular structure, right? So you see that you have the ribbons all packed in together. So whenever you see the ribbons, the helices, those are indicative of secondary structure. But if you have multiple of those that are folded together, that tells you it's tertiary structure. Okay? So you have tertiary structure of a transmembrane protein. Okay? Parts of that protein itself have to be hydrophobic, and parts are hydrophilic. What part of this protein do you think is hydrophilic? A, B, C, or D? A and D. Why? Because it's by where the water is, right? So, but based upon this protein structure, based upon the structure of the phospholipid bilayer, the protein that is going to be part of that membrane has to have aspects of that are hydrophobic and other aspects that are hydrophilic. Okay? Where was this protein most likely produced in the cell? In what part? The ER? The rough ER, right? Let's be specific about that. Okay? There's a difference in terms of the face of phospholipids as well. You have a cytoplasmic side versus an extracellular side, right? So you look at the different faces, different sides of those. All right? Is there a difference between cells, say mammalian cells, your cells, their membrane, and say cells that bacteria or archaea, or a cell that can live in a hot spring, such as Old Faithful Geyser, right, where it's 98 degrees Celsius pretty much all the time, and we went and you looked for cells, and lo and behold, cells are there. Do you think there's a difference in the membranes of those cells? Amelia's nodding her head yes. Why? What does that environment say about the membrane itself? What has to happen? Think about the structure of the membrane and what the environment is going to dictate is going to happen. Is there going to be a difference in structure? Do you all agree? Yes, there's going to be a difference in structure. How is it going to differ? In its carbohydrates? Yes. What about the makeup of the phospholipids? Is that going to change? Yes or no? Yes. All right. Because a higher temperature means that you could put more saturated fatty acids in there. Right? Different types of the components of the phospholipids change. Right? And this actually changes in your cells that your cells will respond to differences in temperature. You can make a minor difference in your cells in temperature and actually change the way change what phospholipids are produced to produce membranes, right, to help with fluidity and etc. These proteins that are found in the membrane, right, we have integral and peripheral proteins. They have 6 major functions, right? They function in transport. That's a very big one. Move things in and out, okay? This is an example of active transport, where you see up here. This is active transport. Why? What's happening? ATP is usually being utilized. Okay? So this one right here is active transport. It's active because ATP is being utilized. And what else is present there? How is the motion of that red molecule going? Up. It's going up. It's going outside. Okay? It's going from where to where? How many red molecules are on the inside of the cell? 1. How many red molecules on the outside of the cell? 5. So what's the concentration of molecules outside? High. What's the concentration of molecules inside? Low. All right? Typically, active transport moves molecules against the concentration gradient. All right? You all know what a concentration gradient is? Yes. Who doesn't? And needs me to explain it more? I can. All right, good. Okay, that was good. Concentration gradient. It's simply, put it simply, it is a number or an amount of molecules. Okay? Over time, there's a certain amount of molecules on the inside, on one side of the membrane, and a certain amount of molecules on the outside. Typically, what's the motion that's occurring? If you look in the blue up here, typically, the blue molecules move from an area of high to low. And they will do that passively. That occurs all over the place all the time, right? In chemicals, in gases, in water, in solids, right? You put Kool-Aid in water, right? A tablet of Kool-Aid or powdered Kool-Aid, if you don't touch the water, it's going to eventually become one complete homogeneous solution, okay? That's diffusion. That is a passive process. Okay? That is moving from an area of high concentration where all that powder is to low where it's not until it reaches equilibrium. Right? Diffusion. In this case, this concentration gradient, there's more molecules outside than inside, but they're still moving the molecules out to the cell, even though that's against where they're going to, where they want to go. In order to do that, they have to use ATP. Okay? In the blue, you see high to low. High outside, low inside. And that protein is functioning as what? As a channel. All right, this is an example of what we call facilitated diffusion. Okay? Either in facilitated diffusion, molecules move along their concentration gradient from high, an area of high concentration, to the area of low concentration. In this case, with the addition or with facilitated by a protein. In this case, it's a channel. All right, so this molecule, blue molecule, just moves through passively as high to low. Okay? In other cases, it could be a binding issue, right? This molecule binds and then moves in based upon high to low. Questions on that type of transport protein? We'll get into more transport in a little bit. Intracellular membranes can be, or integral membranes can be enzymatic, right? Example being ATPase, or this is also could be whatever a enzymatic function can be, but it's a function of the membrane and it's important for the membrane itself. This could be important for signaling, right? A signaling molecule attached to the cell then does some type of enzymatic activity on the intracellular side, which causes signaling to occur inside the cell. Okay? There's attachment to cytoskeleton and extracellular matrix. What group of proteins do we know fits that category, that function? Fits in here. Links between, give you another hint there. What group of proteins fits in that? This is your integrins. Okay. Cell-cell recognition is another function of integral membranes. Okay. Integral membranes have these carbohydrate. This glycoprotein has a carbohydrate chain attached to it. Carbohydrate chains are important in identifying what is your cells versus what is someone else's cells or something else. Okay? How many of you know your blood types? How many of you are A? How many of you are B? AB. O. Okay. Those of you who have O, you lack these carbohydrates on your red blood cells. Okay? That means that you can give blood to whoever else because they're not going to recognize it as foreign. Right? Your cells won't recognize a foreign blood cell because the carbohydrate's not present. Those of you who have blood type A, where are you again? Okay? You have a certain carbohydrate chain on this that identifies your red blood cells as A type. Okay? And if you give your red blood cells to an O-type person, that O-type person will recognize those cells as being foreign and produce a reaction against it. So cell-cell recognition, right? The recognition of self versus non-self. That's what these carbohydrate chains, glycoproteins, are important for. We have intracellular joining. What is this? Or intercellular joining. Give me an example of E. We went over them on Friday. Junctions between cells, tight junctions, desmosomes, right? Gap junctions, they're all parts of that intracellular joining, modulated by or regulated by integral membrane proteins. And then finally you have signal transduction, single molecule receptor, and then movement of the signal from the outside to the inside. Okay? Example of that is nerve to muscle contraction. Okay? This is the beginning of what we call, this is related to transport. Right? We're going to start transport, but it's also an example of sidedness of proteins, of the membrane. Sidedness. Okay? If a receptor, if an integral membrane protein gets put out to the plasma membrane, right? And you see these little Y things here. There's transmembrane glycoproteins, right? These little carbohydrates, these little Y-looking green things. What happens if they're flipped? If these are not out like that, but instead they're out like this. Is that going to change the function of the cell or of that protein? Yes. Those glycoproteins their function is for cell-cell recognition. In order for that to happen, they have to be on the extracellular surface. Right? They have to be facing extracellularly. Okay? Not on the extracellular side, not the cytoplasmic side of the membrane. Because the phospholipid bilayer is not different, right? It's phospholipids, one side or the other. Okay. But the way the proteins are arranged, outside versus inside, there's a sidedness, or what we call a face, to the different sides of the phospholipid bilayer. And that directly relates to their function. Okay? So you have cytoplasmic face and extracellular face, one side versus the other. Now, when you get into upper level classes, you will actually see that the makeup of the phospholipids can change depend upon extracellular versus intracellular space. All right? Depending on the face of the membrane. All right, so diffusion. This is diffusion. Before we get into diffusion, let's talk about transport. Transport, because that's a big function of proteins in the plasma membrane. The function of the plasma membrane is to regulate this transport. We describe the plasma membrane as being what? Give me the description of the plasma membrane, Sabrina. Selectively permeable, right? Which means that some things go in and some things do not. Selectively permeable, or selective permeability. Okay? Some things in, some things move in easily, some things do not. What is an example of something that would not move in easily? From lab, polysaccharides. Why? So here's your bilayer, right? This being your hydrophilic. I'll just put philic. This area in here is phobic. We know it's hydrophobic. Okay? Here's your tails. I'm not going to draw them all out, but you know what I mean. Okay? What molecules don't move in easily? Someone said starch. Why not starch? It's too big. All right, so we could say something that's too big. What else? Polar. Meaning it has a charge. Does not move in easily. Okay? Why doesn't the polar molecule move across the membrane easily? Because it has to cross a non-polar region, right? This middle portion in here, which is hydrophobic, is non-polar. Polar molecule goes through, it's going to run into problems. Yes? The heads are charged, right? So the heads are charged. They're out here, though, right? They're in the hydrophilic portion. So they're attracted to that polar molecule, right? So a polar molecule will stay on one side or the other, but it won't move past because of the non-polar boundary in between. Okay? So we have a size exclusion. We have a charge exclusion, right? That's pretty much the two things that we want to move in, right? How does the cell get past that? How does the cell get charged molecules in? Because these charged molecules, ions, have to move in. Both, right? Both sodium and potassium ions have to move into cells in order for nerve impulses to go, and for muscles to contract. Okay? So this has to happen. So how does a cell get by that? And we do that by proteins. All right, so we have a protein here. And we showed two examples of it, a protein channel and an active transport. All right, a protein will regulate movement of these molecules into or out of the cell. Okay? So transport across a selectively permeable membrane, right, can occur in a couple different ways. Right? First, if things move across easily, like a lipophilic molecule, right, which is or lipid soluble molecule, so it's lipid. It will move across the nonpolar region easily, such as testosterone or estrogen, which are cholesterol-based. They're lipids. They move across the membrane easily. So how does that transport occur? It occurs passively, no use of ATP, and usually by diffusion, by the rules of diffusion, moving from areas of high concentration to low concentration. So here you have high concentration, over here you have low. So the net movement of molecules is going to be from high to low until it reaches equilibrium. All right? And once equilibrium occurs, then there is movement of molecules in both directions. Right? So the net movement is 0. Okay? And this shows you examples of both 1 solute and 2 solute. What is a solute? Something that's dissolved into solution. What's a solvent? Something you use to dissolve the solute, right? Paying attention. Okay? So we have diffusion as a process. Right? So we have transport processes. Let's do this. Transport, and we have passive, which is diffusion. Can you give me another passive process of transport? Osmosis, which is simply diffusion of water. Right? And then another passive process. Facilitated diffusion, which we talked about. Right? So it's diffusion. Facilitated diffusion is diffusion from high concentration to low concentration, but with the assistance of a protein. Okay? And then active processes, we have active transport. Again, protein involved, right? But it requires ATP. And then we have bulk transport, which is endo and exocytosis. Those are the transport processes that cells do. Okay? Questions on transports, the processes. We're going to go over. This is an example of showing you osmosis. All right? Depending upon, if you understand osmosis already, great. However you understand it, fine, perfect, awesome. Okay? If you have trouble with osmosis, okay, the way I like to help people understand it is to find water moves based upon its concentration. Okay? This is diffusion. Osmosis is diffusion of water. All right? So wherever there's a high amount of water, where is water going to move? Away from high to low. High amount of water to low amount of water. Opposite to that, if there's a high amount of water, how much solute is there? Low, right? Less. If there's a low amount of water, there's higher amount of solute. If you like to think about the movement of water based upon the amount of solute, by all means go for it. Okay? I do not want to confuse anybody more than you may already be confused. Okay? I like to clarify it by, if you know where there's a high or low amount of water, you can follow where water is going to go. All right? So let's look at this first one. It says lower concentration of solute, higher concentration of solute. So which means lower concentration of solute means what's the relative amount of water? High amount of water, right? Look at the dispersion of molecules. All right, the green molecules up there. There's more water than there is solute, correct? All right, so now higher concentration of solute on the other side means there's going to be lower amount of water, correct? So what's the net movement of water? From this side to this side, okay? Until it reaches equilibrium. Oh, that's not good.